Okay, welcome back to part 3 of chapter 19 on the blood. We'll have the rest of we just have to finish the rest of the types of white blood cells, talk a little bit about platelets, and then we'll end with the process of clotting. So we're almost done. The next 3 types of white blood cells, after we finished talking about neutrophils, is actually in much lower in amount normally. Eosinophils is one of those 3 types that makes up only 2 to 4% of circulating white blood cells. Now, what distinguishes them, eosinophil, from other white blood cells under the microscope is that they have these red-colored granules that exist in their cytoplasm along with a bilobed nucleus. So when you look at their cells under the microscope, their cell would usually appear a lot more reddish in color when compared with other cells. And that's why on the model in lab, the one with the red dot background, that's the one that is eosinophil. Okay, so there are these little features on the red and on the white blood cell that you can recognize which one are they. Okay. Now, they can attack objects, coated with antibodies, of course, via exocytosis of toxic compounds. Now, these are type of special cells that if they need to fight against larger pathogens, of course, our body doesn't only get invaded by microscopic things. Okay, it's not just bacteria and virus. Sometimes we're attacked with multicellular parasites. Okay, like tapeworm, etc. So those, or little other worms that live in the body that is not supposed to be in there. So those kind of creatures are a lot bigger than a cell like this that's circulating around the body. So they cannot do any engulfment or phagocytosis with these multicellular parasites. So instead, they're acting like little soldiers that will release these toxic chemicals, and those chemicals ingested by these multicellular parasites can then, be poison for that parasite and end up killing them. Okay, so that's what eosinophil specializes for, is the attack of multicellular parasites via exocytosis of those poisonous or toxic compounds. Okay, all right. So they're also very effective in allergic reactions as well, and they are attracted to the site of injury when we have allergic reactions. Okay, and on the same note, we already talked about what basophil does last semester with inflammation, like mast cells. Okay, their distinguishing feature physically under the microscope are that deep purple granules, okay, in the cytoplasm of these cells. Okay, so their background is usually very blue and very dark purple, and they're in charge of releasing histamine and heparin, as we have mentioned last semester. Histamine is for dilation of your blood vessels, for more blood flow and more permeability, and heparin will prevent abnormal blood clotting other than the site of injury. Now, monocytes is the third type of white blood cell that doesn't exist in great amount. Okay, they are the largest in size, and that's why you can kind of see it relatively speaking by looking at the red blood cell compared to the whole white blood cell. If you look at those in relative to all the other white blood cell, monocytes are pretty big. Okay. They have a large oval kidney bean shaped nucleus. So they have this big purple part of it, okay, that serves as its nucleus. In the end, what monocytes ends up doing is they would turn into macrophages. And macrophages are aggressive type of phagocytes that we'll talk more about when we get to the lymphatic system or immune system. They will release chemicals that attract neutrophils, monocytes, and other phagocytic cells and fibroblasts for healing of a wound and producing scar tissue that we talked about last semester. Okay, and last but not least, the second most abundant type of white blood cells are called lymphocytes. Okay, Lymphocytes are the smallest of all white blood cells. Okay, if you compare again with red blood cell size, they are pretty almost close to the same size of a red blood cell. And their distinguishing feature physically on the microscope is that their nucleus is very round and large and almost take up the entire space of the cytoplasm. You can only see a small amount of cytoplasm around the cell, kind of like a halo around the nucleus, okay? Whereas the other ones, you can see a lot of cytoplasm space along with the nucleus like that. Lymphocyte, very easy to distinguish. They have that huge nucleus and a small little halo of cytoplasm around it. Now, even though these white blood cells are small, they are very powerful fighters against pathogens. We'll talk more about them when we get to the immune system chapter, but they are subdivided into 3 functional classes. One class is called T cells or T lymphocytes. They are responsible for cell-mediated immunity that we'll talk about in the immune system, but it's a specific defense mechanism, meaning that we're going to talk about non-specific versus specific attacks. And for T cells, or all lymphocytes, they do specific attack, which means one specific type of T cells will fight one specific pathogen. Let's say this is for strain flu A, okay? So that particular T cell only attack flu strain A, okay? And then another T lymphocyte will then attack flu strain B, okay? So they're specific for their particular pathogen that way. And why they're called cell-mediated immunity is because T cells will attack the cell themselves, okay? They don't release anything that to attack the foreign invader, but they will attack the cells themselves. B cells, on the other hand, or B lymphocytes, are responsible for what's called humoral immunity. Humoral usually means something to do with fluid. So these are the guys that are responsible for producing antibodies that's going to help either mark the pathogen as foreign so other cells can fight them and eat them up, or the antibodies will destroy the cell itself. So activated B cells will actually differentiate into a specific group of soldier. We call it an army of soldier cells called plasma cells, where they would divide in massive amounts to make large and huge amounts of antibodies so you can quickly fight the pathogen specifically. And the last type of lymphocyte is called natural killer cells. And as the name itself speaks for how scary it is a very powerful type of cell. They usually carry out our immune surveillance for detection and destruction of any abnormal cells, not just pathogens, but if it's coming from our own cell that has gone bad, for example, cancer cells. Okay, natural killer cells are the only type of immune cells that can recognize those potentially cancer cells. Okay, all right. And, when you have usually white blood cells is in small amount, but not to the point where you don't have any. When you have not enough amount of white blood cells, that is called leukopenia. Remember, penia is when you don't have enough, okay? Not enough. And that's why when we talked about bone chapter last semester, we said osteopenia, which means you don't have enough bones. So leukopenia means you don't have enough white blood cells. And you can't have too much white blood cells either. They're supposed to be in that small amount, okay, in your body. So excessive numbers of white blood cells detected in a person's blood is called leukocytosis, which actually can indicate 2 things. If it's moderate increase in white blood cell, that's normal in a person that has an infection, or if they're sick with a cold, or other things, okay? But too much, we're talking extremely large amounts of white blood cell, can actually be an indication of leukemia, which is cancer of the blood, where they're just making a huge amount of white blood cell due to the fact that those white blood cells those stem cells itself that makes the white blood cells has become cancerous itself. All right. So, and we just And just as we had talked about erythropoiesis for red blood cells, we have leukopoiesis for white blood cells. Leuko, again, for leukocytes, poiesis for generation of, so it's generation of white blood cells. And again, they all start with, here's that chart I was talking about that does a good breakdown of what hematopoietic stem cells will do, okay? They're also called hemocytoblasts, but I don't know, I got used to the fact that they're called hematopoietic stem cells, so I like to go with that, but you are free to remember it as hemocytoblasts too. But to me, hematopoietic stem cells speaks for what they do more so than the other name. Okay, so for leukopoiesis, you still start with hematopoietic stem cells, and then you differentiate into myeloid stem cell or lymphoid stem cell. And myeloid stem cells would then differentiate into all types of blood cells down here other than lymphocytes. Lymphocytes actually form from lymphoid stem cells in a process called lymphocytopoiesis. Okay, lymphocytopoiesis is illustrated by this right here. So this is lymphocytopoiesis. This right here is what we talked about as erythropoiesis, and this right here then is leukopoiesis, okay, where myeloid stem cells can further differentiate into your basophil, eosinophil, neutrophil, and monocyte. I'm not going to ask for these different things in between, okay, so don't worry about those. And lymphoid stem cells usually migrate to your peripheral lymphatic tissue. It doesn't stay within the bone marrow for lymphocytopoiesis to be completed. Lymphoid stem cells will actually break off from the bone marrow and then travel into your peripheral lymphatic tissues such as your thymus, spleen, and lymph nodes, which we'll talk about in the lymphatic system chapter, and they will mature within those organs. T cells specifically will mature in the thymus, as we have mentioned in the endocrine chapter, for the function of thymus and why thymus makes the hormone thymosin for helping with the maturation and development of those T cells. Okay. All right, so last but not least, okay, that's all we're going to talk about for white blood cells, because we're going to talk more about them in the lymphatic system. Okay, so last but not least, we have the last type of cells within the blood's form elements called platelets, or the fancy name for them is called thrombocytes, because they make thrombin. Okay, well, that's why they're called thrombocytes. They're disc-shaped cell fragments that play a major role in vascular clotting system. They're, again, continuously replaced just like all the other blood cells. If anything, they survive even less than red blood cell, only 9 to 12 days before they're removed by phagocytes and will be replenished instead. Again, they have three main functions, all of which has the ultimate goal of stopping from bleeding, and the process of stopping from bleeding is called hemostasis. Don't confuse this word with homeostasis. Again, it's kind of like melatonin and melanin. They're off by a few, one or two letter, but they mean completely different things. So homeostasis is regulation of stable internal environment. Hemostasis is hemo, okay? Hemo, remember I said that usually deals with blood. Stasis, stopping, stabilize, okay? So stabilize blood flow. And that way you're stopping bleeding, abnormal bleeding from happening. All right. So platelet will function to release chemicals, enzymes, and whatever other factor that is important and critical for initiating the whole clotting process, which we'll talk about in a bit. They will form a temporary patch called the platelet plug to plug up that area where the blood vessels has been torn or damaged so that you don't have any more blood cells leaking out of that area. And they also can reduce the size of the break in the vessel using actin and myosin. Okay, and we'll talk about that. So their production, as you saw from the previous diagram, also comes from hematopoietic stem cell. Okay, so if we go back to this diagram here, you have hematopoietic stem cell, myeloid stem cell, and then they will become what's called megakaryocyte. So megakaryocyte is the precursor cell for platelets. So this I want you to remember, okay? Megakaryocyte, and then what happens is you see this is a huge cell with lots of little stuff, okay? So these little stuff is actually going to break off and form your little platelets at the end. in the process of producing those platelets, okay? So the process of forming platelets is called thrombocytopoiesis. No surprise there, thrombocyte generation, okay? Thrombocytopoiesis. And like I said, they come from precursor stem cells called megakaryocytes. Okay, so again, the process of stopping bleeding, establishing a network or framework for tissue repair is called hemostasis. And this process is divided into 3 phases that we're going to talk about in detail: vascular, platelet, and coagulation phase. And you have to know these 3 phases, okay? So the first vascular phase has the same, as the name suggests, has to do with blood vessels. That's why it's called vascular, okay? It has to do with blood vessel, and usually bleeding happens because a blood vessel was cut or torn or ruptured in some way, some form of another. So the first thing that needs to happen is make that opening smaller so you can control the bleeding very, very quickly. And this happens via a process called vascular spasm, which decreases that diameter of the cut into a smaller hole to really slow down or even stop the bleeding process. Endothelial cells from the walls of blood vessel actually releases a hormone called endothelins. Okay, so endothelins released by endothelial cells. Okay, easy to remember in that case. And endothelins are the hormone that will promote that contraction of vascular spasm to cause the blood vessel to contract and make that opening a lot smaller, and hence less blood will leak out. In addition, these cell plasma membrane also become sticky, and that will help to seal off the cut and promote platelet attachment. So it's kind of like not only are we making the hole smaller, but we're also making these area a little bit more sticky, like glue, so that when platelets come by and flow by, they're going to stick onto that surface and eventually will close up the wound, or close up the opening. So that's what this second phase is about. So the second phase of hemostasis is called the platelet phase, where you begin with first platelet adhesion, which is the whole point of why we needed to make that plasma membrane sticky on the endothelial cells, because then the platelets will stick to those sticky areas and starts aggregating there, okay? So once one sticks, they will actually recruit more platelets, and they start piling on top of each other in a process called platelet aggregation. Just like this, they start piling on top of each other. I'll show you an animation video that illustrate this process very well. And they form a platelet plug in the end by piling on top of each other, and eventually they will seal up that opening with that platelet plug so blood doesn't leak out from the blood vessels anymore. And remember, we talked about this whole process a little bit when we were talking about positive feedback from AMP1. How one platelet getting stuck to the area enhances more platelets to come. It's an enhancing process. That's why it's a positive feedback loop. And usually this platelet phase begins very quickly after an injury, about 15 seconds, to minimize the amount of bleeding that can occur. So you can tell right away when some blood vessel is torn, the first thing happens, vascular phase, vascular spasm. Then the next thing that quickly happens is platelet aggregation, platelet plug formation in this platelet phase. And once they arrive at the injury site, platelet will become activated. Okay, what does activation means? It means they will change their cellular morphology and become more spherical and develop these cytoplasmic processes, and they kind of extend like little aliens toward nearby platelets, where they would start attaching and making communication processes with other platelets. Okay, so when kind of like, we humans can link arms with each other, that's what those processes are kind of like doing with the platelets. They can link arms with each other and form this huge pile of platelets that can be stuck in that area to seal up the wound. And once activated, platelets can be start to release all these different things on this list. I don't expect you to remember all of these, but at least remember calcium ion is a critical component for the process of clotting. Okay, so and Under normal conditions, prostaglandins should also be released so we don't start forming blood clots everywhere, kind of like how heparin works, okay? It makes sure that platelet plugs only happen around the injury site because if blood starts clotting or platelets starts aggregating anywhere they want, wherever they want, we'll be in trouble because all these blood clots will start forming and they will block blood flow, and we don't want that. We only want it to be restricted to the injury site itself. The last phase is most difficult phase to remember because it is more complex and there are multiple parts involved. So, but you have to remember these pathways. Okay, it's called coagulation phase. That's the last phase of clotting. And this is the phase that will finalize the process of producing what's called fibrin that reinforces the platelet plug. Because just having the platelet plug there is not enough. You need to kind of like how you have to, if a stuffed animal has been ripped open, you put the cloth over it, but the cloth doesn't stay there on its own. You can try to put glue, but it won't stay that well. You kind of have to sew it out with strings, okay, to really make sure that piece of cloth stays patched on the stuffed animal. Same thing here, okay? Just having that platelet there, platelet plug, okay, that doesn't make sure it stops it from moving around. So that's why you need these strings in forms of fibrins that will kind of reinforce the platelet in that particular area, make sure it doesn't float away or dissolves away. Okay, so this phase rely on the presence of multiple clotting factors which are responsible for activating the production of fibrin. Okay, and what are some of these clotting factors is what are missing for individuals with hemophilia. You've heard of people with the genetic disease called hemophilia where their wounds can't clot, and what they're missing are these some of these specific clotting factors in their blood. All right, so something that seems so simple, just to produce strands of protein that closes up the wound, actually requires 3 separate pathways to work together. You have the extrinsic pathway, which is this one right here, extrinsic pathway, which mainly involves factor 3 first, which then binds to calcium, and which then binds with calcium and factor 7 to form an enzyme complex that will activate another enzyme called factor 10. And then the intrinsic factor here will reinforce that process, but they will start with different factor. They will start with factor 12 right here, which will then bind with calcium, and factor 8 and 9 as well to form the enzyme complex that will ultimately activate factor 10 as well. Okay, and both of these pathways will have a common goal of activating factor 10, and factor then goes on to activate conversion of prothrombin to thrombin. And from thrombin, they will be able to convert fibrinogen. Remember those proteins we talked about in the beginning of the chapter as one of the plasma proteins that floats around in the blood. That fibrinogen will then be triggered by thrombin to turn into fibrin, okay? And fibrin are those little string I was talking about that will produce the final blood clot, okay? So pretty complex, okay? But I try to narrow it down by these number. So 3, 7, and then it goes to 10, okay? The intrinsic pathway go from 12, 8, and 9, and then they make 10, okay? Both require calcium, and then they would have to be activating prothrombin to thrombin, fibrin, which turns fibrinogen into fibrin, and fibrin will start making these strings that will weave in and out of these platelet plug to ensure that it stays in place. And this is a video that will, animation video that will illustrate this whole process, that I'm not just making this up or making it more difficult for you. You'll see it in this video.
At the site of vessel injury, the first platelets arrive to start sealing the wound. Simultaneously, the coagulation cascade with its various coagulation factors is activated. This involves 2 pathways. the extrinsic and the intrinsic pathway. Extrinsic activation begins with now exposed molecules of the vessel wall, such as tissue factor, which forms a complex with factor 7. Finally leading to the activation of factor 10. This factor 10A is the point at which the extrinsic and the intrinsic pathways of the coagulation cascade meet. The intrinsic pathway consists of various coagulation factors activating each other in a chain reaction. At its end, a complex with an additional cofactor is formed. This complex now activates factor 10. Since the two pathways merge at the level of factor 10A, this factor has a pivotal role in the coagulation cascade. Further down the cascade, factor 10A, in combination with 5A, activates thrombin and induces the so-called thrombin burst. One molecule of factor 10A can catalyze the formation of 1,000 molecules of thrombin. These large amounts of thrombin cause the further activation of platelets and the enhanced formation of fibrin.
There's
Fibrin then forms strands, making up the mesh that stabilizes the platelet plug in an arterial clot and holds together the red blood cells in a venous clot. It can be concluded that the central role of factor 10A in the coagulation cascade makes it a viable target for therapeutic intervention in pathologically altered blood coagulation.
Okay. Let's continue. So that's a wonderful animation that illustrates that whole process we're just talking about, and we're almost done. Since the abnormal blood clotting can actually lead to very dangerous situations like heart attack and stroke if we start forming blood clotting everywhere, and that's probably why that whole last coagulation phase is so long and complicated, to ensure that there are multiple checking systems in order for blood clotting to occur. If blood clotting was an easy mechanism, then it is more likely that some things going wrong can lead to abnormal blood clotting. And hence, there are also things that the body does to control blood clotting to ensure that we don't create dangerous situation of abnormal blood clotting. Normal plasma will contain several different types of what's called anticoagulants that will be sure that will inhibit clotting, like antithrombin. Okay, remember thrombin is the whole in the end, sort of the overall thing that will cause formation of fibrin. So there are antithrombin 3, for example, that's formed as an anticoagulant. And we have already mentioned basophil and mast cell will release heparin, which usually will serve to inhibit abnormal clotting and works by accelerating the activation of this particular antithrombin 3. Thrombomodulin can also inhibit clotting factors. Prostacyclin is actually a part of the family of prostaglandin we talked about earlier that can inhibit platelet aggregation at inappropriate location. Okay, and all three pathways of blood clotting will require calcium and vitamin K. Okay, both of these must be present in the liver in order for us to synthesize 4 of those clotting factors we talked about, including prothrombin. And the last 2 steps of clotting process, of course, once the wound is healed, you have your basal, remember all those basal cells we talked about that goes underneath and starts to push the clot up. So that's got to be the last process of this clotting mechanism, is to remove the clot. Okay, the clot has to go through clot retraction, which removes the fibrin that the cell works so hard to produce, but we need it in order to stop the bleeding at the moment, so that we don't bleed to death, and also protect our body cells from infection of other pathogens. Okay, close up the wound so there's no opening for those pathogens to try to enter in. So an enzyme called plasmin will be activated to digest those fibrin strands in a process called fibrinolysis, and that's when the clot will then be able to remove, okay? It's kind of like working so hard to make these fibrin, but then at the end you still have to remove them in order for the clot to go away, and then your new epithelial cells are formed to close up the wound. Okay, and that's all I have for you for chapter 19. We'll go over the practice questions and then we'll be done. Okay, which of the following is not a major function of the blood? They do not store calcium ions. Everything else they do. What is the primary difference between plasma and interstitial fluid? Plasma contains significantly more protein than interstitial fluid. Okay. Which of the following does not lead to decrease in hematocrit? Dehydration. Okay, remember dehydration causes an increase in hematocrit because you lower the amount of plasma level, which makes your foreign elements a lot higher percentage compared to the whole blood volume. There are 280 million hemoglobin molecules in one red blood cell. A single hemoglobin carries how many molecules of O2, which means one single red blood cell would carry how many oxygen molecules? One hemoglobin has 4 slots for oxygen, so that means one single red blood cell carries about 1 billion oxygen molecules. And when we go through these practice questions, be aware of these type of question. You see how it's kind of application of what you know and not directly testing for definitions. And we had definitions back in A&P 1 more often, but in A&P 2 there are a lot of these mechanistic type of question that's more therapeutically related or physiology related. Okay, so be sure to make good notes about those physiological effects of the body. Why is it important that when iron binds to oxygen and that this is a temporary and reversible interaction? We talked about that, is to enable it to dissociate easily into tissues. We don't want it to permanently bind. Under normal condition, what is the fate of globular proteins of recycled hemoglobin molecules? They are disassembled into their component amino acid because it's asking for the fate of globular proteins. If it's talking about heme, then it's different. Okay, if it's talking about alpha and beta chain, then it's different. Okay. Which condition below would stimulate release of EPO? We had that 4 things, and all of these was one of those 4 things that we talk about that can release or trigger the release of EPO. Which characteristic of blood contribute to its high viscosity, interaction with proteins, lots of foreign elements in there, and water molecules within the plasma itself? All of these kind of interact with each other in a sense that contributes to that viscosity. Which organ produces all the albumin in the blood circulation? We talked about that being the liver. Andrew has blood type AB positive. What type of blood can he receive from a donor? He's AB positive, so he is a true universal recipient, which means any blood type he can receive from. He's a lucky guy because his body does not make antibodies against A, B, or Rh antigen. Why can't a person with blood type A safely receive blood from a person with blood type B? Because blood type B will agglutinate in the recipient's blood vessel and plug small vessels to vital organs. So that's why we cannot just give the wrong blood type to the wrong person. Why is it unlikely that the antibodies in the plasma of type O donor would cause a transfusion reaction in type A recipient? Because the plasma portion of donated blood is so tiny when compared with the volume of the plasma in the recipient's body. This was what I was talking about, how there's only small amount of the antibody within the blood that's donated, that it doesn't matter. Okay, it's more, what matters more is the antigen that's on top of the red blood cell, whether it will trigger an immune reaction within the recipient's immune system. Why is cross-matching performed even though both donor and recipient are known to be the same blood type? Is because there can be still other potential cross-reactions, surface antigen, actually more than 48 of them, other than A, B, and Rh available. What type of white blood cells found in greatest number in an infected cut? We talked about neutrophils being the most abundant. Which of the following is not a characteristic of all white blood cell? They are not all phagocytic. Okay, remember we talked about how some use exocytosis of toxin, some attacks in other formats. Okay, so not all of them are phagocytes. Monocytes transform into what kind of tissues? Monocytes become macrophage. They start with the same letter, so that's how you can easily remember that. And they will become phagocytic and release chemicals that attract other neutrophils. SAM has infestation of large parasitic worm, which in the following would happen. Large parasitic worms, so eosinophil is the one that's going to be activated. What contributes from the extrinsic and intrinsic pathway to help with enzyme complex necessary to begin a common pathway? 3 and 7 come from extrinsic pathway. 8 and 9 come from the intrinsic pathway, and both of them mix factor 10, which then cause prothrombin to thrombin, and then from thrombin we're able to turn fibrinogen into fibrin. Okay, and there will be a case study on blood. Okay, and that's all for this chapter.